Well, good morning, good afternoon, good evening, everyone, and welcome to the world's most exciting classroom event. My name is Joe Kourounis. I'll be your host for today. We are jumping into our fourth episode geared towards classrooms in Australia, New Zealand, and the surrounding areas. And we have a lot to cover in our hour together today. So the very first thing I want to do is update everybody with our map. So if you visit DARWIN200.com, you can find our map. It's interactive. You can see where the ship, the Oosterschelde, is at any given time. You can see photos from along the way. And what's really cool, I don't think we've showed this off yet, is we have a bird survey ongoing that's covering the entire 2 years of the voyage. So if you click this little bird icon, it will share the lists of what we're seeing as we're going. So if I click over here, you can see that there was a count 8 red-tailed tropic birds. We had some pitcairn reed warblers. So you can get an idea of the bird life that we're seeing along the way, which is pretty darn cool. And then, of course, we have our pictures as well. I think the most exciting thing you're going to notice is that the ship has moved. After about a month hanging out on the North Island of New Zealand, undergoing some maintenance, having some young Darwin leaders on board the final week, we are finally moving, and we are moving fast. We're already, looks like, halfway across the Tasman Sea on our way to Sydney, Australia. Let's take a look at a couple of these pictures here. Let's see what we got here. A nice view there. Out on the bowsprit, shooting back, looking at the ship. Very cool. Got a few more pictures back here. Looks like we have some bird life. Oh, that might be an albatross. I'll have to check in. That looks like it could be an albatross. That's very cool. So we got some great shots there. Awesome stuff. All right, let's come back from that screen share. And today we are going to get to head live on board the ship because it's moving. There's passengers on board. There's researchers on board. There's crew on board. So let's get on board right now, the Oosterschelde, and let's see who's there waiting for us. All right. Hey, George. Hey, Josh. How you doing?
Hi. All good. Great, Joe.
All right. Great to see you guys. How are things going? How's the sailing?
Yeah, going well. We had a bumpy first couple of days, but it's calmed down a lot now. So we've got some nice wind in the right direction. We've been doing a lot of going in the wrong direction. We've got a bit of time. So to keep sailing, we've just been moving with the wind. But it's finally blowing the right way now. So we're heading straight towards Sydney.
And look to increase today, even more wind.
Yeah.
Perfect. Okay, well, let's meet the two of you because we have not connected yet with this group of classrooms. So Josh, why don't you start off and introduce yourself?
Yeah, sure. So hi, everyone. As Joe said, I'm Josh. I'm one of the full-time camera guys for DARWIN200. So me and the other guy, Tom, who's not on board at the moment, we sail on the ship as it goes around the world, and we document all the life on board, all the wildlife we see. We take all the photos, we make all the videos, and then when we're in port, we work with the Darwin leaders and we make the films for the Darwin leader projects with them. So yeah, I've worked for the project basically since the beginning, since Tenerife, and I've had the chance to go to some really cool places and film some things I never even dreamed that I would film, and meet some awesome people along the way.
All right, very cool, Josh. I'm going to ask you a couple questions in a moment, but George, why don't you take a moment, introduce yourself?
Thanks so much. My name is George. I'm from Toronto, Canada. I'm a professional explorer. I'm with the Explorers Club. I'm also explorer in residence with the Royal Canadian Geographical Society. I specialize in extreme places and weather, things like that. And this is my first time on board the ship. I've been here for about a week now on board, and it's been an amazing experience. We've seen some really, really cool stuff. I'm also helping out with some of the research that's being done on board, which I'll talk about more in a bit.
Perfect. Well, Josh, you've been on board for quite a while. You and I have met up in a few areas around the world. Can you share maybe a highlight or two from the journey so far? Something you did, something you saw, something you filmed?
Yeah, sure. I mean, the first one that always jumps to mind for me is, if it's a sailing leg that I've done, it would be sailing through the Chilean fjords. That was absolutely incredible. A lot of the time when you sail on the ship, you sort of leave the port, and then 6 hours later, you don't see land anymore, and then you don't see land again until 6 hours before you arrive. But sailing through the Chilean fjords, we had amazing views every day, huge mountains jutting out of the water. We managed to sail right up to a huge glacier. I think it was the 4th biggest glacier in the world outside of Antarctica or something. And then to top it all off, towards the end, we got to spend a few hours in a bay hanging out with a load of blue whales, which was just amazing. But as well as that, I think as far as a location I've been goes, the Falkland Islands really tops it for me, because I'm a penguin man. I love my penguins, and there's a lot of penguins in the Falkland Islands. So yeah, that was an unforgettable time, and I'm lucky enough to be heading back there again in February. So hopefully more penguins.
All right, very cool. Well, we have some great footage from the Falkland Islands. I'll probably share that in a future event. And of course, we're going back there. So hopefully we can get some of those satellite units out onto the island, maybe even in one of those colonies. That would be pretty cool.
Yeah.
Yeah. All right, George, let's get you in for a little bit. We'd love to hear a little bit about your experience on the tall ship so far. Have you ever sailed on a tall ship before? And then let's get into what you've been seeing.
Yeah, I've sailed in various places around the world. I've sailed around Cape Horn in a small sailboat and spent a lot of time at sea, but never on a ship like this. This over 100 years old, this beautiful schooner. It's absolutely magnificent. So it's a real treat for me to be on board. And there's a lot going on. Let me get my screen sharing here so I can show you some of the stuff that we've got going on. And there we go. There we are. So there I am with the historic flag of the Explorers Club. That particular flag has a lot of history. It's been to dozens of different expeditions all over the world. And it has been on this entire journey. And as a member of the club, it's a real honor for me to be able to hold that and carry that on this leg of the journey. But the main reason why I'm sort of here is doing a bit of research, gathering some environmental DNA. So what is that? Well, we as organisms leave a trail of ourselves behind us wherever we go. We are always having skin cells that are sloughing off, or fish are swimming through the water and there'll be bits of scale and fecal matter and whatnot that come off of them. And if we sample the environment that they live in, we can determine by the residual DNA what creatures have been here recently. And that's what we're doing right now. So here's a sample I took this morning of the seawater off of the starboard side of the ship. And basically what we do is we gather this water and filter it so that we get rid of the water and then keep all of the other bits that happen to be floating in the water. And this includes all kinds of various cells from all of the creatures that are in this area. And then those filters can then be sent to a lab to be processed to find out what creatures have been at this particular location. So every day I'm taking GPS measurements, measuring the temperature and the acidity of the water, and gathering these water samples. I don't know right now what creatures live near where the ship has been going, but we will be able to know once these samples have been analyzed, which is pretty cool. So you don't need to even be able to see the creatures to know that they have been in the area. So it's a really non-invasive, very sort of environmentally friendly way of doing wildlife surveys and biodiversity studies, things like that. Now, I spend a lot of my time chasing extreme weather. I chase tornadoes and hurricanes and things like that. And the weather out here on the ship has been really fascinating to me. It's so beautiful. We get these beautiful sunrises and sunsets. And we've encountered a few different natural phenomena that I kind of want to share with you guys because they're sort of rare but really interesting, and a little bit about how they come about. So here's an example. There's the sails here on board the ship. And the other day we had this beautiful ring around the sun, and that's called a solar halo. And they're actually not super uncommon, just people don't know to look for them. And they typically only appear when you have very thin clouds that are consistent and very high up in the atmosphere. We call these cirrus clouds or cirrostratus clouds. And on a couple of occasions on the journey now, we've seen these. So this one was from about 3 or 4 days ago. And then here we had another one from, I think it was day before yesterday. So I'm aiming right at the sun, and you can see this ring around the sun. And if you were to take your thumb and block the sun with your thumb and then extend your pinky finger, that's about how far away from the center of the sun that halo looks like to us here on the surface of the Earth. And the physics behind it is a little complicated, but I'll simplify it. Basically, sunlight is passing through ice crystals up in the atmosphere. And they're very specific ice crystals. They have to have 6 sides. So hexagonal ice crystals. And by having a plain, flat sheet of these up in the clouds, in these cirrostratus clouds, the light hits them and splits into its various components, kind of like how a rainbow works. That's why you see these colors, these reds and sort of oranges. So it's kind of like a circular rainbow, but instead of using raindrops, it's basically powered by these ice crystals that are high up in the atmosphere. So we've been super lucky to spot a few of those. You can see these wherever you live in the world. They're not dedicated to certain parts of the planet. So if you've got a day that has really high sort of wispy clouds, block the sun with your hand and take a peek up, and you might see some of these. Really, really interesting. Now, 2 days ago, we saw something that really got me excited. There's this phenomenon known as the green flash. And it's really hard to see, but it's one of those things that if you capture it, it's quite special. Mariners and sailors have been talking about the green flash and legends of the green flash. It happens right at sunset, the moment the sun dips below the horizon. Now, we've all seen lots of sunsets, and we know that the sun, when it gets low in the sky, it takes on that orange or yellowish globe, the circle kind of shape. But sometimes it gets a bit distorted as it gets closer to the horizon. And then very rarely, when the sun, just in the last few seconds before it disappears, that yellowish color will turn a bright green, literally for less than half a second. And that's what happened the other day. Here's a zoomed in shot. You can really see that bright green. And the reason why this happens has to do with what's going on in the atmosphere. So as that sunlight is passing through the Earth's atmosphere, it bends a little bit, but not all of the colors bend the same amount. So if you have a layer of warm air over top of a layer of cooler air, the sunlight will bend, and that green color will be the one that we sometimes see on these rare occasions. Here's a photo of one that I took up in the Arctic a few years ago. So I've seen them in the Arctic, and now I've seen them here in the South Pacific. And if you're on the beach and you're watching the sunset, you'll sometimes see the sun take on this weird, distorted shape, kind of looks a bit like a lampshade. And then there in the last few moments before the sun completely fades out, the top will sometimes burst with this bright flash of green color. And it really is an emerald green, but it only lasts a fraction of a second. So here we can see we've got that warm air near the surface. You've got cooler air, which is a bit more dense. And so as the sunlight passes through these different layers, it sort of splits. And then we here at the surface see that green. But please, please don't stare at the sun. Don't look at the sun. Protect your eyes from the sun. It's only in those last few seconds where the sun is filtered by our atmosphere that it's kind of safe to look at the sun in those last few moments. And if you're really, really, really lucky, you'll encounter that green flash. You have to have a very clear horizon all the way out to the western horizon. No clouds, no mountains, no buildings in the way. So the best place to see this is either on a boat or at the beach looking west at sunset. So there's just a couple of examples. There's a really good example of a green flash. That photo is not mine. I wish it was. But there you can really see that yellow disk of the sun in the last few moments of its setting with that little bit of green light that's being bent by our atmosphere, kind of like bending a piece of plastic, and only the green color is appearing at the top there. So yeah, just some amazing stuff that we're seeing out here. I don't know what we're going to see over the next week before we finally make it to Australia. But every day I'm out there looking for interesting things to spot in the sky in between taking those DNA samples.
All right. Pretty cool, George. Those are some great photos. I've got some questions coming in here via the chat. Josh, one for you here. This is from Charlotte. She's wondering, is it ever scary to be traveling on a ship all over the world?
No, not really. I think this ship has sailed for over 100 years and it's not run into any problems yet. And I think the crew and the captain are very capable. And even in the worst weather that we've had, where we've had howling wind and pouring rain and the boat's been going 40 degrees one way and 40 degrees the other way, as long as the captain looks fine, you've got nothing to worry about. And I haven't seen the captain look scared yet, so I think we're going to be okay.
It's a fine ship.
All right. George, a fellow George would like to know how long you've been into exploration.
Oh, geez, since about 1997. So I guess over 25 years now. It started out as a hobby, an interest in science and nature and photography. And then I got really interested in weather and different natural phenomena, specifically extreme natural phenomena. So I started chasing tornadoes, documenting lightning, hailstorms, then got into hurricanes, wildfires, exploding volcanoes, the polar regions, pretty much any extreme place on Earth or any extreme phenomena. So about 25 years, yeah.
All right. Well, I saw this question coming up, and I thought I'd share a quick little clip for the students of you in another environment. And this is a video you sent to me years ago, but it's one of my favorites. So I'm going to play this little clip here.
I'm explorer and adventurer George Kourounis, and when I explore, I do it by the seat of my pants.
All right. So not kidding around there, right inside at the lava lake of a volcano. It's pretty wild.
And just to point out, that's actually not too far from where we are. That video was shot in the island nation of Vanuatu, which is also here in the South Pacific. That was on Ambram Island. We're going away from that right now, but it's not too far away.
All right. So Annabelle wants to know the eDNA. How are you getting the water when the boat is moving?
It's not that difficult, actually. I'm actually using those little test tubes, and I attach a piece of string to it, and I just literally drop it down the side and whoop, grab some water and haul it right up. It's pretty simple.
All right. Josh, Quinny wants to know, you've obviously been on the ship a lot on the ocean. What was the best thing that you found or saw in the ocean?
I think it has to be when you were actually on board, Joe. When it was a night between Peru and Ecuador when we had crazy bioluminescence in the water, and then a pod of dolphins came and joined the ship in the middle of the night, and it was pitch black and you couldn't see the dolphins, but you could see the shape of the dolphins in the bioluminescence with this streak of light behind them. And they stayed with us for the best part of an hour, and I think we went and woke everybody on the ship up,
banging on everyone's door.
Yeah,
that was pretty wild
experience.
And I remember it was almost like a torpedo. You'd see them coming in from further out. You could see that line, that bluey green line through the water as they came racing in. That was
that was really amazing.
That's wild. We are seeing bioluminescence at the stern of the ship every single night, though. So we're not, of course, to the degree that you guys saw, but we're seeing that as well. These dinoflagellates that self-illuminate once you agitate them, which is really interesting.
Yeah, so for our classrooms in North America, we just did an experiment where we grew some dinoflagellates. We're going to have the results pretty soon. I'll tell you, it's pretty beautiful. We'll do this experiment probably in a few weeks with the Australian classrooms as well. George, Bianca wants to know, what made you want to hop on a very old ship?
Sorry, what made me want to
hop on a very old ship?
Oh, I knew that this was going to be a challenge for me because I do tend to get seasick. And spending 2 weeks at sea, not being able to touch land, it came across as a really fun challenge for me. I've known about this expedition for a very long time, and I've been very envious of some of the legs of this journey. And when the opportunity came up for me to do this particular leg, it was like, yeah, I want to do this. I want to see if I can do this. And so far, I've been having a great time. So as a personal challenge, I'm loving it.
All right. One more question for you. This is from Jet. What was the worst hurricane you've ever been in? Josh, I don't think you've been in one yet, but George?
Not yet.
Yeah. Well, for me, the worst one that I've ever been in was Hurricane Katrina back in 2005. I was in Gulfport, Mississippi, and the entire city was breaking apart all around me. There was a casino building that was lifted by the waves and dropped into its own parking lot. So that was very intense.
All right. Very cool. Okay, we are going to let you guys go shortly because I know lunch is coming up. That's an important time on the ship. Oh, those birds. Do you know what species of albatross those were?
Yeah, we've had white, the ones with the nice eyebrows. They're white-capped albatross, and then there's also royal albatross in there as well.
All right.
Yeah, there was
excellent.
a nice day where we had sort of 5 or 6 of them following the boat, close to the boat all day, and we soaked it up because we've seen a handful since then, but quite far away and quite quick sighting. So it was nice to have them for almost the whole day.
All right, very cool. Okay, well, I am going to let you guys go. We didn't go outside today because it's a little bit windy, but we will, in upcoming events, we will explore the ship a little bit. What do you think, George? Can you pop up and just show us outside for a second?
Stand by.
Okay.
I'm going to try, Joe.
Okay.
If we get cut off, it was been a great session, but I'm going to try and stick the laptop outside and give you guys a view of what the ship looks like.
Yeah,
Fingers
it should be
crossed.
good. The Starlink's pretty good up there. It should be okay for just to poke out the hatchway. I'll make it nice and full screen here. There we go. Beautiful. We have some sails up. Oh, you've got the nets up. Very cool.
Can you see that okay?
Yeah, beautiful.
Awesome.
Oh, jealous. I can't wait to catch up to the ship in a little bit.
Thanks,
All right,
guys.
George. Thanks so much, George. Thanks, Josh. We'll see you again soon. Thanks, guys.
Bye now.
All right. Very cool. Hard to tear away from that view. It looks absolutely beautiful. They're having some good sailing. Okay. Well, I see our next guest hanging out backstage. I'm going to bring Dr. Jen Matthews in with us shortly. But before we do, I'd like to share some little clips with you guys from the journey so far, because classrooms in Australia and New Zealand really just started joining us about, I guess this is our 4th event, so about a month ago. But we have done so much since then. We've been sailing for a year and a half. We have crossed the Atlantic Ocean. We've made a complete navigation around South America, to then headed to the Galapagos and all the way across the Pacific Ocean. So the clip that I chose to share with you today is a little bit related to our curiosity of the week, which I'm going to share the answer for from last week near the end. But when we were sailing area of the Pacific Ocean called Whakareva in the region of Tahiti, we did some diving. And we saw some beautiful, pristine coral reefs. These coral reefs were like jumping into the water and being in an aquarium. So much marine life, so much fish. And then, of course, really good, healthy coral reefs have lots of sharks. So I want to share this little clip with you. It's about 3 minutes. And then we're going to meet our next guest speaker for today's event. So bear with me for a moment while the video file kicks in. And here, there it is. Here it comes. All right. So that is the clip I wanted to share with you this week. In Tahiti, beautiful, healthy coral reef, tons of marine life, and a real good sign of a healthy coral reef, lots and lots of sharks. Now, you wouldn't recognize Maria there. Maria has joined us in our last 3 events, but from Indonesia with another Darwin researcher, Ranva, and they are on a cave diving expedition. So you'll recognize Maria from the last 3 weeks, but she was there doing coral transects for us. And so she was in, you saw her as the diver with the blonde hair in some of those clips. So there's Maria in another environment. We saw her in the cave system, and then that's her in the, in the ocean. Very cool. All right, let's get our next speaker in. We're going to continue talking a little bit about corals. And we are going to bring in coral biologist Jennifer Matthews. She's joining us from Sydney, Australia. Her research tries to understand the cellular processes that underpin coral health and function. So she wants to better inform conservation activities to help safeguard the world's coral reefs. I'm going to bring in her in with us right now. Hey, Jen, how are you?
Hi, I'm well, thank you. How are you?
I'm good. It's great to see you. We've got you in Australia. I'm all the way here in Canada. The ship is coming your way, and we've done a few of these events together, but I'll actually get to see you in Australia in about a week. That'll be pretty cool.
Oh, that's excellent. Yeah, I wish I was actually on the Great Barrier Reef, but Sydney is just as magical. We have some corals here as well. But thank you for inviting me to the world's most exciting classroom.
All right. Well, we're thrilled to have you. I'm sure there'll be lots of questions that will come in during the event. So any classrooms tuning in, put questions in the chat as they come to you. And Jen, I'll let you take over for a little bit and share with us a little bit of what you get up to.
Fantastic, thank you. So I am the Deputy Team Leader of the Future Reefs Group at the University of Technology, and we are a group that basically researches coral health in order to protect them for future generations to enjoy. So I'm going to talk to you a little bit today specifically about one particular life stage of corals, and that's coral babies. So before I start, I wish to acknowledge the traditional owners of the land and sea country on which UTS stands, as well as the Irukandji and the Gunganji people, where a lot of the research that I took where they're from. And I want to pay my respects to their elders past, present and emerging, and recognise them as the first scientists and knowledge keepers of the land and sea, and welcome any First Nation people here today. So coral reefs are foundational ecosystems. They have immense ecological, economic and cultural value. And healthy reefs actually can support about 7,000 marine species. That's about 25% or a quarter of all marine species. They provide billions of dollars and millions of jobs. They help protect our coastlines and are actually an important source for new medicines. So medicines for Alzheimer's and cancer have actually been found from corals. And here in Australia, we are home to the iconic Great Barrier Reef, the only living structure in the world that can be seen from space. But unfortunately, without help, coral reefs like the Great Barrier Reef may not survive this century. For example, increasing sea surface temperatures is causing the catastrophic loss of corals in our lifetime and on our doorstep. And this has led to international calls to increase our knowledge of what determines their health and also their resilience to climate change or environmental changes. And hopefully with this knowledge, we can better inform conservation and restoration strategies and help to safeguard the coral reefs and the services that they provide. And here's an example of some of the ways that we help reefs. Whether it's buying time for them through conservation and restoration, monitoring their health through to help inform policies, or researching what makes a more resilient coral. All of these are really important mechanisms to help saving our reefs, or as I said, at least buy time for them while we get our act together on climate change. And that's what I've dedicated my life to doing, whether it's research in the field or in the lab, or it's outreach or education. I hope to make a meaningful contribution to the protection of coral reefs. So I'm not sure if you know, but what is a coral? Do you think it's an animal, plant, or just a rock? Well, it's actually a trick question because it's all three. There is the animal that looks and is very similar to a jellyfish, but they also have inside their tissues these tiny single cell plants or algae. And just like you and I, corals need nutrients to thrive and survive. They need food. And for the coral animal, they can get the food from the seawater using their tentacles, like you see here on the left. But they can also get a lot of their nutrients from that algae in their tissues. And it also gives them those amazing colors that you see, especially when you put them under UV light, like I'm showing here. They become fluorescent, like little neon animals. And being an animal, they also reproduce. Corals actually produce millions of babies each year. And it's in a magical event that's actually a few days after full moon in spring. And the next one, would you believe it, is in about 10 days. So if you live near a reef and you're able to get out there, it's definitely worth a visit. But unfortunately, less than 1% of these coral babies ever survive. And this is mainly thought to be because they just don't have the energy. They don't have enough food to make it through to adulthood. Again, just like us humans, food is a basic need, and coral baby nutrition, we know so little about it. And that's kind of the gap I'm trying to currently solve. I'm essentially researching whether we can help coral babies by feeding them with a coral baby food. And if we can improve their survival and the number of those millions of babies that make it through to adulthood, that will increase the diversity and the number of corals on reefs. And as Darwin described, this is essential for organisms to evolve to new environments. So you can see some little coral babies swimming around here. And I was very fortunate to receive funding from the Pure Ocean Foundation to investigate this. Essentially, we capture some of these coral babies during these mass spawning events, like the one in a few days, and we feed them with something called lipid nanoparticles, which are just a fancy way of saying balls of fat. And essentially, with these little balls of fat, we help to nourish them. And we're doing this both in the lab as well as directly on the reef. And we hope that these chubby, nourished little corals can then be grown to help restore and rebuild degraded reefs. And so I'd like to take you through one of our recent trips where we actually went out to the Great Barrier Reef to Moore Reef with Reef Magic, an ecotourism operator on the Great Barrier Reef, to feed some coral babies. And talk about a lab with a view. This is how we set up our lab before the corals started to spawn. And we deployed underwater these kind of tents, essentially, that collect the coral babies as they are released into the water after the sun sets. They release the eggs and the eggs float up into that little container at the top, and then we can bring those back to the lab. And so now it's spawning time. Corals release these egg and sperm bundles into the water as well. So there were some that obviously we didn't put tents over all of the corals. So some of them just released into the water. But the ones that we were able to collect, we brought them back to the lab and we fertilized them in the lab. And the picture on the right there is actually freshly fertilized coral babies. They are in a stage that we call the prawn crisp stage or the prawn cracker stage. Over the next few days, we then monitor their health and their numbers and make sure that they're doing well. And this is until it takes about 2 days for them to form a mouth. Once they've got a mouth, then it's time to feed them, essentially. And we either do this in the lab, as I mentioned, or we put them into nurseries, which are just made out of floating paddling pools with the bottom cut out of it. And we use these nurseries to help provide them with a nice, safe place that they can become juvenile or swimming baby corals before we feed them. It means that they're not going to be eaten by fish or predators, and they've got a nice, safe place to grow up. And then it's time to feed them. So this is some of the food, the baby food that we gave them. And we did this in collaboration with the Gunganji and Irukandji people and the sea rangers on the Great Barrier Reef. And you can see here, I am feeding the coral babies in their little nurseries out on the reef. And because the little balls of fat, they're all biodegradable. So even if the larvae don't eat them, the babies don't eat them, then the fish can, and they get a nice supper too. So one of the really interesting and surprising results that we found was not just that by feeding them can we enhance their survival, but we can also make them swim better. And so you can see here a little video of some coral larvae that we didn't feed on the left. They're swimming nice and slowly, nice and carefully, but when you feed them, they do little coral zoomies. They go a lot faster and a lot further. And this is really important when we're thinking about things like reef connectivity. This is how reefs between longer distances apart are able to be connected through reproduction. For example, if a larvae is able to swim better and further and faster, and then in a healthy reef could help to restore a neighboring degraded reef just by having better swimming larvae. And as I mentioned, we also supported their survival. So when we didn't feed them, we saw a survival rate of about 37 to 45%, but by feeding them, that rate went up to about 70 to 88%. So quite a substantial increase. But what we also found was if we exposed them to heat stress, like increased sea surface temperatures, we had a survival rate in unfed larvae of between 30 to 39, but when we fed them, that survival rate was still up and around 60 to 65%. So we're not only improving their survival under normal conditions, but under predicted climate change conditions as well, which is really important when we're thinking about how corals into the future and how reefs are going to look into the future. So once they're ready and once they've been fed, then it's time to what we call settle them, which means giving them a chance to become baby or young adults. They settle onto tiles. They really like to grow on rocks. So you can kind of see the little ones here. They're seeking a little place to grow. Find a nice place to grow. This is on a boat, so it was a bit rocky for them, but they did eventually grow on these tiles, and we can put them back onto the reef so that they can mature into adult corals. And then when they're big enough, then they could even help to restore entire reef systems. We can select areas that have been degraded from maybe cyclone events or from recent impacts from maybe boats or weather events, that kind of thing. And with any luck, we can help to regrow and restore some of these degraded reefs. And these kind of projects are not going to save reefs. I'm not saying that this is the answer to save reefs, but unless we take serious action on climate change, then all they're going to do is buy time for reefs. But they do highlight how some basic biology, like food, are really, really important avenues to explore when we're thinking about protecting reefs in Australia and beyond. And so thank you so much for your time. I'm very, very grateful for the opportunity to talk to you a little bit about some of the research that we're doing. And if you're interested in studying coral reefs, then yeah, please come and give me an email, shoot me an email. I always love talking about corals.
All right. Jen, thank you so much. What a cool presentation and really cool research. I really like seeing the coral with the zoomies after they had something to eat. Very cool. All right. Well, we're going to do some questions in just a moment. I have some questions coming in via the chat, both the private chat and the YouTube chat as well. But before we do, I want to play a quick Kahoot quiz together, which we do every week here. So I am going to quickly share my screen. I'm going to bring the Kahoot up front and center. I'm going to make sure that it starts okay. That looks good. All right. You should see it now. So, Kahoot.it, that's what we want to know, Kahoot.it. The pin is 213456. You could join as a classroom. If you're joining from home, you could scan. You can see there's a little QR code up there in the corner. You could scan that QR code and it will bring you into the Kahoot. Or if you can't get onto technology, join in anyways and shout out your answers and see how you do. So we'll give a moment to see if we get a couple groups in here with us, and then we'll go ahead and play our Kahoot one way or another. Yeah, and while we're waiting, Jen, maybe ask a couple of these questions here from the chat.
Yes.
Let's see. Okay, so Ebony would like to know when you started working with coral.
So I started working with corals. I went on holiday to Thailand for just a 2-week holiday. It was meant to be a very quick holiday. I ended up staying 4 years because I went diving for the first time, and I think I was about 20. And yeah, I went diving for the first time and saw corals, and they were actually going for a stress event at the time. They were these really neon pinks and bright oranges and fluorescent yellows. They were honestly, so it's just remarkable. And I thought, wow, they're so colorful. What are these things? And I came up and I asked my instructor, and they said that actually they were really stressed, and that color was a stress response. And I thought, if something could look that beautiful when it's stressed, imagine what it could look like when it's healthy. And so I stayed in Thailand and founded the marine conservation organization Big Blue Conservation in Koh Tao. And so that's when I started working with coral, and it's been a love affair ever since.
All right, very cool. Let's squeeze in one more, and then we'll go to the Kahoot. Charlotte would like to know the deepest place you've ever dived with coral.
Oh, corals like that I study with the algae, they actually only live down to about 30 meters, because that's as far as the light can penetrate into the ocean. You can get some deep sea corals. They can go all the way down to hundreds of meters. I've never dived with any of those. Not the easiest place to access as a diver, but certainly you get on tropical reefs, you can get reefs down to about 30 meters, and that's quite typical.
All right. Well, let's play some Kahoot. It's going to load up here. It's going to count in. We've got a few groups in with us, which is great. And let's see who comes out on the podium. So our first question is a multiple choice. What services do coral reefs provide? Coast protection, medicines, nursery habitats for fish, or all of the above? So you have 4 options there. What services coral reefs provide? Coast protection, medicines, nursery habitats for fish, or all of the above. Okay, good job, crew. All of those are right, but the best answer there is all of the above. Let's take a look at our leaderboard. We got the Royal Lark in first place. True or false coming your way? Great Barrier Reef is one of several natural-made objects that can be seen from space. So the Great Barrier Reef is one of several natural objects that can be seen from space. So made by something natural. All right, good job, crew. It is the only one. It's not one of several. It is the only thing made by living organisms that can be seen from space. Let's go to our next question. That puts the Space Sphinx into first place, and we are going to another question. This is a multi-select, so you have to pick everything you think is right. So coral is a plant, rock, bacteria, or animal. So you have to pick every one that you think is right there. So that's your hint. It's more than one. You have to click as many that you think are right there. So plant, rock, bacteria, animal. You've got about 8 more seconds to get your answer locked in. 2 and 1. All right. I don't think we've had one of those multi-select before, so that was a tricky one. There was lots you had to pick there. But like Jen told us, plant, rock, animal. We did not talk about bacteria. Royal Lark is on fire. They are back in the lead. Let's go to a true or false. Corals produce millions of babies each year. Is that true or is that false? Corals produce millions of babies each year. All right, that is absolutely true. One more question coming your way. I think one more question. Royal lark still in the lead. True or false? 10% of coral babies survive to adulthood. Is that true or is that false?
Wow, that's a blue coral. That's pretty cool. I've never seen that before.
Yeah, just one of those preloaded Kahoot images.
Yeah, nice.
All right, that is false. 1%, Jen, is right. 1% make it past that young stage. All right, we're going to take a look at our leaderboard. In third place, we've got the Lucky Possum. In second place, we've got the Space Sphinx. And holding down that top spot, all right, the Royal Lark. If you are the classroom that contains the Royal Lark, I need you to send me a message to classroom@darwin200.com. Classroom@darwin200.com. We have a gift card that we will send your way. Classroom@darwin200.com. All right, I'm going to turn off that music. We're going to come back from that screen share and squeeze in a couple more questions here. This one's from YouTube. Jen, what are your expectations, or maybe science's expectations, for coral reefs in the next 10 years?
That's a really good question. There's an optimistic and a pessimistic probably expectation, but I think I am a cautious optimist that we are likely to still have coral reefs. They may just look rather different. So unless we do something about climate change, then they're going to continue to degrade. We're going to see entire species being lost, especially those branching ones that look like little trees. They're going to be the first to go. And that's really unfortunate because they often have the ability to house little fish. They like to live in the nooks and crannies. So without those beautiful branching corals, then our reefs are going to look very different. They'll look a little bit quieter. There won't be as many fish. Turtles, sharks will start to leave to find food elsewhere. So unless we do something about climate change and we can help to restore some of the degraded reefs and protect some of those coral species that are most vulnerable, then they're going to look very different.
All right. Let's see. Harry wants to know the most important thing about your job. What do you think is the most important thing about the job that you do?
Probably this, one of my favorite things to do, and one of the most important things is sharing knowledge. So there's only so much that you can do as a scientist, and if you keep it to yourself, then what's the point? The really important thing is that we share that knowledge and inspire future generations like yourselves to become coral biologists. And even if you don't live near a reef or you don't have the opportunity to visit coral reefs, then there's still a lot that you can do for marine conservation, like selecting seafood sustainably, participating in other ecological monitoring or sustainability programs, or when you get the chance, and voting correctly, those kind of things, or voting for sustainability, those kind of things.
All right. Final question for you, Jen. How many people do you work with on your research team?
Oh gosh. So there's currently about 20 students and researchers in the Future Reefs group, but I work with a variety of different people. I work with a number of traditional owner groups, as well as ecotourism groups and people in the industry. So it's not just I'm alone doing this and sort of trying to save corals. There is a huge network of people trying to improve coral reef status. So yeah, definitely, I would say in Australia, there's probably thousands of people dedicating their lives to protecting coral reefs in some way or form.
All right. Well, Jen, thank you so much for being with us tonight. Thanks for taking us into the world of baby corals. You're doing really important.
You're very welcome.
It was great to be able to share in a little bit of it this evening. And we're really excited to catch up with you when we're in Sydney, and maybe we'll even get you live in person on one of our world's most exciting classroom events. That would be
I would love that. That would be wonderful. Yes.
All right.
Have a great day. Thank you so much.
Thanks, Jen.
Bye.
Okay, well, to wrap up for today, we have our experiment and our curiosity. So 2 weeks ago, we had an experiment looking at osmosis with gummy bears. So let's play the results And then I have a class who sent me in some cool pictures. So we'll share those pictures, excuse me, after we play the results of the experiment.
Welcome back. Let's look at the results of our osmosis experiment. Looking at the 3 gummy bears, I can see that they're very different shapes and sizes. They've definitely changed overnight. These have been in the solutions for about 24 hours, so it's now time to measure and weigh the gummy bears once more. Well, looking at the 3 beakers, I can see that the gummy bears are very different sizes. Let's start with the one in the concentrated salt solution. I'll pick him out carefully with these tweezers and put him on the scales. So, when we put him in, he weighed 2.2 grams. He now weighs 1.5 grams. So I'll write that down. Let's also quickly measure him. He is, wow, he's 17 millimeters long. And I'll get his widest bit, 9 millimeters wide. We'll compare the results later. Now let's look at the gummy bear in the weaker salt solution. Very carefully, I'll bring him out and put him on the scales. Wow, he weighs 4.8 grams. So I'll write that down here. And measures 28 millimeters long by 14 millimeters wide. Now let's look at the gummy bear in the pure water. There's no salt in this beaker at all. Let's pop him down there. Wow, he weighs 5.7 grams. And he measures 36 millimeters long and approximately, it's a bit difficult measuring these squishy gummy bears, but we'll do our best, 16 millimeters wide. Now let's look at the results. So the gummy bear in the really concentrated salt solution, he weighed 2.2 grams when we put him in that solution, and now weighs 1.5, so he's lost 0.5 grams. And he measured 21 millimeters long and 12 millimeters wide. He now is just 17 millimeters long and 9 millimeters wide. So he's actually shrunk both in terms of size and in weight. Now let's look at the gummy bear in the weaker salt solution. When we put him in there, he weighed 2.3 grams, but now weighs 4.8 grams. So he's expanded and is now heavier. He also is larger. When we put him in, he was 22 millimeters long and 11 millimeters wide. And he's now 28 millimeters long and 14 millimeters wide. So he's definitely grown in both weight and in size. But the biggest change definitely has been the gummy bear that we put in pure water with no salt at all. When we put this guy in, he weighed 2.3 grams, and he now weighs 5.7, so he's nearly doubled in weight. And he's also expanded in size. When we put him in the water, he measured 22 millimeters long and 12 millimeters wide, and now is 36 millimeters long and 16 millimeters wide. So he has also expanded significantly in size. Did you work out why the gummy bears have changed in these different ways? Well, let's start with the one in the concentrated salt solution. This one has shrunk. This is because the salt concentration in the solution was so much greater than the internal salt concentration inside the gummy bear. So the water within the gummy bear has been drawn out, shrinking its overall size. This is exactly how many cells work. If you put a cell in a really concentrated salt solution, the water will be withdrawn through osmosis, shrinking the cell overall, exactly like the gummy bear. Well, these two have expanded in size. The gummy bear in the weaker salt solution has absorbed a lot of water. The osmosis effect wasn't quite as strong, so the water wasn't withdrawn from inside the gummy bear, rather the contrary. Water was absorbed from the solution into the gummy bear. And of course, that's even more the case in the one in pure water because there's no salt at all. So this one has expanded and expanded to its maximum size. This experiment demonstrates how cells respond through osmosis. Most cells will shrink just like the gummy bears if they're in really saline conditions, because due to osmosis, the water within the cell is withdrawn, shrinking the cell overall. Similarly, cells in less saline environments or pure water can absorb the water through osmosis, so they'll often expand. How did your results compare to mine?
All right, so let's take a look. We got a great submission here from a classroom in South Australia. Let me see if I can get that shared up on the screen here. Yep, there we go. So we'll take a quick look at those results. All right, so if we scan through, we can see some of the students there. You can see them making their solutions. You can see they're weighing. There's Stu there in the background. They're watching the instructions. Very cool. Leaving it for 24 hours. Good job. Look at those gummy bears swelling up. Very, very, very cool. All right. Thanks so much to the crew. We might just have a gift card to send your way for sending in that great experiment example for us. We have a brand new experiment that you can try over the next 2 weeks. Let me get it queued up here. This one is about bioluminescence. So it's very timely that Josh talked about bioluminescence being one of the favorite things he saw on board the Oosterschelde.
Welcome back. During the last few weeks, the team aboard the DARWIN200 ship have reported seeing lots and lots of bioluminescence in the waters around the vessel as they've been cruising along the coast of Brazil. Bioluminescence is when organisms release chemicals that produce light. This creates amazing flickering glows. They can be yellow, blue, green, sometimes other colours as well. Different organisms produce bioluminescence for lots of different reasons. Some deep-sea fish produce bioluminescence to see and to attract and hunt prey. Other organisms, such as tiny dinoflagellates which float in seawater, they produce bioluminescence as a form of defence. The spectacular glow can startle or disorientate predators, or cause the predators to really stand out in the dark water and perhaps make them more visible and more vulnerable to larger predators. Other organisms, such as fungi, also glow, but are a bit more mysterious, and the reasons for this are less well known. Today's experiment explores bioluminescence as a form of defence. To undertake this experiment, you need a beaker with water. You can also use a jam jar or a glass cup. You need a fluorescent yellow marker pen, a torch or flashlight, some scissors, and some paper towel or tissue paper. Very carefully, with the supervision of an adult, cut off the back of the marker pen, and out pops this felt section with the ink inside. Pop that into the beaker and let the ink slowly dissolve into the water contained within the beaker. It should take about 30 minutes or so. When all the ink has been dissolved from this part of the marker pen, you can take that out and put it on the paper towel. The ink can stain things, so definitely put it somewhere safe and sound where it won't cause a mess. Then you're ready to experiment with your solution. Turn off the lights if you can, and then with the torch or flashlight, have a little look. As you move the light around, what happens? Can you see a big shine, a bit like those flashing glows from the bioluminescent animals? Imagine you're a predator in the deep sea. How would a bright bioluminescent flash of light make you feel? Would it make you afraid? Would it startle you? Would it scare you off? Or would it make you even more hungry and more aggressive? Email in photos of your experiment along with your thoughts to classroom@darwin200.com and let us know how you think you would react to this bright bioluminescent glow. We've got 3 £50 Amazon gift voucher prizes to give away. Join us in 2 weeks' time to find out the answers.
All right, so there we go. 2 weeks to give that a try in your classroom. and send us in some pictures or video clips of your students giving it a go. So we're going to wrap up today with our curiosity of the week from last week. Before I play that video, I had some great guesses come in. So we knew it was used to call something, to attract something. And we had guesses that range from cats to turtles, to bush babies, to beagles, to woodpeckers, to sea lions, snakes, birds. The guesses went on, but unfortunately, although they were all great guesses, none of them were correct. So let's take a look at what the actual use for that caller is. Here we go.
This week's curiosity was this object here. This is a very difficult curiosity to guess. This is a shark caller. People across different parts of the Pacific use these to catch sharks. Fishermen go out on canoes, put it over the side of their boats and rattle it really loudly. And amazingly, the sound that the rattle makes actually mimics that of a fish in distress, which the sharks are attracted to eat. But of course, in this case, the sharks come up to the boats and the fishermen then catch the sharks and take them back to the island communities where they feed their families. So a bit of a tricky curiosity of the week this time. This is a shark caller from the Pacific.
All right, and we have a brand new curiosity for you to try and decipher this week. Remember, there's 1 week to try and decipher the new curiosities.
This curiosity is this object here. Can you guess what this is? We've actually got an episode coming up specifically about this object. So hopefully you can work out what it is. I'll give you a clue. It grows on a plant. Good luck and submit your answers to info@darwin200.com.
All right. So just to correct that, that should be classroom@darwin200.com. So classroom@darwin200.com, 1 week to send in your best guess for what you think that is. Okay, well, that was another busy event. We ran a couple minutes over, but that's because we just packed so much content into this hour-long World's Most Exciting Classroom extravaganza. We're going to sign off for today, but before we do, a reminder that we'll be meeting again next week. We have another exciting event coming your way. And of course, a huge shout out to our sponsors, partners, supporters, without whom we wouldn't be doing this World's Most Exciting Classroom events. We wouldn't be sailing around the world, training young conservationists, or undertaking really important research projects. So I hope to see more classes joining us next week, and we're going to sign off for now. Thanks, everyone.